Hello everyone, we hope that you guys already read today's topic. Most of us will sleep for well over 175,000 hours in our lifetimes. This extraordinary commitment of time implies that sleep fulfills a critical biological function. But what is it? And what about dreaming? Why do we spend so much time dreaming? And why do we tend to get sleepy at about the same time every day? Answers to these questions await you in this chapter. We welcome you to chapter 13, where we'll be talking and discussing sleep, dreaming, and circadian rhythms. For this chapter, we'll delve into the following main topics: stages of sleep, why do we sleep and why do we sleep when we do, effects of sleep deprivation, circadian sleep cycles, 4 areas of the brain involved in sleep, drugs that affect sleep, sleep disorders, and lastly, effects of long-term sleep reduction. Stages of sleep. Many changes occur in the body during sleep. This module introduces you to the major ones. 3 standard psychophysiological measures of sleep. There are major changes in the human EEG during the course of a night's sleep. It was discovered that although EEG waves are generally high voltage and slow during sleep, there are periods during sleep when they are similar to the low voltage fast waves of wakefulness. In the 1950s, it was discovered that rapid eye movements occurred under the eyelids of sleeping subjects during the periods of low voltage fast EEG activity. In 1962, Berger and Oswald found that there was a dramatic decline of EMG activity in the muscles of the body core during these same sleep periods. Since these 3 discoveries, electroencephalogram or EEG, the electrooculogram or EOG, and the neck electromyogram or EMG became the 3 standard psychophysiological bases for defining the stages of sleep. The figure depicts a volunteer participating in a sleep experiment. A participant's first night of sleep in a laboratory is often fateful. That's why the usual practice is to have each participant sleep several nights in the laboratory before commencing a sleep study. The disturbance of sleep observed during the first night in a sleep laboratory is called the first night phenomenon. 3 stages of sleep EEG. According to the American Academy of Sleep Medicine, there are 3 stages of sleep EEG. Examples of these 3 stages are presented in figure. Just before a subject falls asleep, the EEG is typically punctuated by bursts of alpha waves, or the large amplitude regular 8 to 12 Hz waves indicative of relaxed wakefulness. There is a gradual increase in EEG voltage and a decrease in EEG frequency as the person progresses from stage 1 sleep through stages 2 and 3. When a subject falls asleep, the EEG progresses in sequence through initial stage 1, stage 2, stage 3, and stage 4. Initial stage 1 sleep EEG is low voltage, fast activity similar to but slightly slower than that of wakefulness. Stage 2 sleep EEG is of higher voltage and slower than stage 1. Its most obvious characteristics are K-complexes, or a single large negative wave followed by a single large positive wave, and sleep spindles, or a 1 to 2 second waxing and waning burst of 12 to 15 Hz waves. Stage 3 sleep EEG is defined by the predominance of delta waves, the largest and slowest EEG waves ranging from 1 to 2 Hz. Once the sleeping subject has gone through initial stage 1 to stage 3, he or she goes back through the stages to stage 1 EEG. When the subject returns to stage 1 EEG, it is referred to as emergent stage 1 EEG because it emerges from the other stages. The rest of the night is spent going between emergent stage 1 through the stage 3 sleep and then back again. It is important to distinguish between initial stage 1 EEG and emergent stage 1 EEG because only emergent stage 1 EEG is associated with REMs and lowest levels of muscle tones in core muscles. It is important to distinguish between initial stage 1 EEG and emergent stage 1 EEG because only emergent stage 1 EEG is associated with REMs and low levels of muscle tone in core muscles. The stage during which emergent stage 1 occurs is often called REM sleep or paradoxical sleep. It was initially considered a paradox that subjects sleep, while their EEG suggested that they were awake. The progression of EEG stages changes during a typical night's sleep. Each cycle is about 90 minutes long. As the night progresses, less time is spent in stage 2 and 3, more is spent in REM sleep. And there are brief periods of wakefulness which are normally forgotten in the morning. The sleep associated with emergent stage 1 EEG is often called REM sleep after the associated rapid eye movements, whereas all other stages of sleep together are called NREM sleep or non-REM sleep. Accordingly, initial stage 1, stage 2, and stage 3 sleeps are sometimes referred to as NREM 1, NREM 2, and NREM 3 respectively. NREM 3 is often referred to as slow wave sleep or SWS after the delta waves that characterize it. REMs, loss of core muscle tone, and a low amplitude, high frequency EEG are not the only physiological correlates of REM sleep. Cerebral activity like oxygen consumption, blood flow, and neural firing increases the waking levels in many brain structures, and there is a general increase in the variability of autonomic nervous system activity. Example, in blood pressure, pulse, and respiration. Also, the muscles of the extremities occasionally twitch, and there is almost always some degree of penile or clitoral erection. REM sleep and dreaming. The discovery of REM in 1953 by Kleitman and his colleagues led to the obvious hypothesis that REM sleep periods were periods of dreaming. This hypothesis was confirmed by waking subjects up during various stages of sleep and asking them if they had been dreaming. About 80% of awakenings from REM sleep led to dream reports, and only 7% of awakenings from non-REM sleep stages led to dream reports. In the years since this discovery, it has become apparent that dreaming during non-REM is more common than previously believed. Antidepressant drugs can reduce REM sleep without affecting dream recall, and cortical lesions can abolish dreaming without affecting REM sleep. Testing common beliefs about dreaming. The correlation between REM sleep and dream recall that Kleitman and colleagues discovered provided them with an opportunity to test some common beliefs about dreaming. The following 5 beliefs were among the first to be addressed. First, are external stimuli incorporated into dream sequences? Yes, dripping water into subjects occurred in 14 out of 33 dream cases. Second, do dreams run in real time? Yes. Subjects awakened 5 or 15 minutes after the beginning of a dream could guess the correct interval on the basis of the contents of their dreams. Third, does everybody dream? Yes. Even people who claimed that they did not dream had normal amounts of REM, and they reported dreams if they were awakened during REM, although less frequently. Are penile erections indicative of dreams with sexual content? No. Penile and clitoral tumescence occur during all dreams, regardless of sexual content. Fifth, are somnambulism and sleep-talking the acting out of dreams? No. Sleep-talking can occur during any stage, and sleepwalking usually occurs during slow-wave sleep. Interpretation of dreams. The Freudian theory of dreams holds that dreams represent unacceptable or often sexual wishes, and was based on the science and beliefs of the 1890s. It has no support today. One widely held alternative to the Freudian theory of dreams is Hobson's activation synthesis hypothesis. Hobson has proposed an activation synthesis theory of dreaming based upon the idea that the information passed on to the cortex during REM is random, and dreams are the cortex's effort to make sense of these random signals. You might liken this process to what happens when you stare up at the clouds and happen to see faces or figures among them. The clouds are randomly patterned, but your brain is trying to its best to make sense of the random pattern. So what do you see when you look at these clouds? Now that you guys have been introduced to the properties of sleep and its various stages, the focus of this chapter shifts to a consideration of two fundamental questions about sleep. Why do we sleep, and why do we sleep when we do? Two kinds of theories of sleep. Two types of theories have developed to account for why we sleep: the recuperation theories and the adaptive theories. The essence of various recuperation theories of sleep is that being awake disrupts homeostasis in some way, and that sleep is required to restore it. This is the way that most people think about sleep. According to the adaptive theory of sleep, sleep is not a response to an internal imbalance. Sleep is an adaptive response that evolved to conserve energy and to protect organisms from mishap and predation. The urge to sleep evolved to be greatest during the night for animals that do not see well in dim illumination. The Circadian theory of sleep considers sleep to be like sexual behavior. It is adaptive, and there is a strong drive to engage in it, but its purpose is not to correct an inner deficiency. Comparative analysis of sleep. All mammals and birds sleep in a fashion similar to humans. Fish, reptiles, amphibians, and insects also go through periods of inactivity similar to sleep. The comparative investigation of sleep has led to several important conclusions. Let's consider four of these. First, the fact that most mammals and birds sleep suggests that the primary function of sleep is not some special higher-order human function. The evidence is strongest in species that are at increased risk of predation when they sleep, like antelopes, and in species that have evolved complex mechanisms that enable them to sleep. For example, some marine mammals, such as dolphins, sleep with only half of their brain at a time so that the other half can control resurfacing for. It is against the logic of natural selection for some animals to risk predation while sleeping and for others to have evolved complex mechanisms to permit them to safely sleep unless sleep itself serves some critical function. Second, the fact that all animals sleep suggests that sleep is not uniquely human, but has evolved for some sort of important physiologic reason. For example, suggestions that sleep helps humans reprogram our complex brains or that it permits some kind of emotional release to maintain our mental health are improbable in view of the comparative evidence. Third, differences in sleep duration suggest that it is not needed in large amounts. Many animals sleep only 2 to 3. Horses and many other animals get by quite nicely on 2 or 3 hours of sleep per day. Moreover, it is important to realize that the sleep patterns of mammals and birds in their natural environments can vary substantially from their patterns in captivity, which is where they are typically studied. Fourth, sleep duration seems to be correlated with how vulnerable a species is when it sleeps. Predators tend to sleep longer, whereas prey species sleep for shorter periods of time. For example, zebras must graze almost continuously to get enough to eat and are extremely vulnerable to predatory attack when they are asleep, and they sleep only about 2 hours per day. In contrast, African lions often sleep more or less continuously for 2 or 3 days after they have gorged themselves on a kill. Effects of sleep deprivation. Interpretation of the effects of sleep deprivation. The stress problem. One way to identify the function of sleep is to determine what happens when a person is deprived of sleep. You need to be cautious about interpreting sleep deprivation studies since when most people are deprived, they do so because they are under stress. Therefore, it is difficult to separate the effects of sleep loss from the effects of the stressful conditions. Predictions of recuperation theories about sleep deprivation. The recuperation theory predicts, first, that long periods of wakefulness will result in debilitating physiological deficits. Second, that these deficits will grow steadily worse as sleep deprivation continues. And third, that after the sleep deprivation has ended, much of the lost sleep will be regained. Two classic sleep deprivation case studies. Let's look at two widely cited sleep deprivation case studies. First is the groundbreaking study of a group of sleep-deprived students. Second is the bizarre case of Randy Gardner. First, the case of the sleep-deprived students. A group of sleep-deprived students exhibited the following features. During the first night, subjects felt very tired and usually felt an attack of sleep drowsiness between 3 and 6 a.m. Those subjects that continued in the study continued to exhibit a wave-like increase and decrease in sleep around the same time every night. Second, the case of Randy Gardner. In a separate case, Randy Gardner wanted to break the world record of 260 hours of conscious wakefulness. He managed to stay awake for 11 days. When he finally went to sleep, the first night he slept 14 hours. Thereafter, he returned to his normal pattern of 8 hours. Experimental studies of sleep deprivation in humans. There are many studies in which subjects have been totally deprived of sleep for several days. These studies have not confirmed the predictions of the recuperation theory. No marked physiological disturbances other than increased sleepiness have been discovered in sleep-deprived subjects. Sleep-deprived subjects often display mood disturbances and problems maintaining vigilance. After 2 to 3 days of sleep deprivation, subjects experience microsleeps, or the 2 or 3-second periods during which subjects remain sitting or standing, but their eyelids drop and they are unresponsive to external stimuli. Sleep-deprived subjects appear to perform poorer on cognitive abilities that require the executive function of the prefrontal cortex. Adverse effects on physical performance have surprisingly been inconsistent. Sleep deprivation studies with laboratory animals. Sleep deprivation in lab animals has suggested that sleep plays a critical role in the well-being of an organism, as several days of total sleep deprivation has led to subject mortality. However, these data should be interpreted with caution, as the sleep deprivation paradigms used in these studies appeared to severely stress the animals involved. REM sleep deprivation. Because REM sleep is associated with dreaming, there has been much interest in the effects of the selective deprivation of REM sleep. There are two major effects of REM sleep deprivation. First, REM deprivation is enforced by waking subjects up each time that REMs begin to occur. As a period of REM deprivation progresses, subjects must be awakened more and more frequently to prevent them from having periods of REM sleep. After REM deprivation is curtailed, REM rebound is often seen. Subjects get more than their normal amount of REM sleep for the next 2 or 3 nights. One of the main challenges for anyone suggesting that REM sleep is critical to normal functioning must explain why tricyclic antidepressants, which block the REM sleep at common therapeutic doses, have produced none of the adverse psychological side effects attributable to REM deprivation in patients who have taken them regularly for years. One current theory, which is the default theory of REM sleep, posits that REM exists simply because it is difficult to maintain a non-REM state. When an organism has bodily needs like hunger or thirst, it wakes up, but when these needs do not exist, the organism maintains a wake-like REM state rather than struggle to maintain a non-REM state. Sleep deprivation increases the efficiency of sleep. A key feature of much of the work on sleep deprivation is that it increases the efficiency of sleep. For example, deprived subjects display more slow-wave sleep stages. This stage seems to play a restorative function. Because this is such an important finding, let's look at 6 major pieces of evidence that support it. First, although people regain only a small proportion of their total lost sleep after a period of their sleep deprivation, they regain most of their lost slow-wave sleep. Second, after sleep deprivation, the slow-wave sleep EEG of humans is characterized by an even higher proportion of slow waves than usual. Third, people who sleep 6 hours or less per night normally get as much slow-wave sleep as people who sleep 8 hours or more. If individuals take a nap in the morning after a full night's sleep, their nap time EEG shows few slow waves, and the nap does not reduce the duration of the following night's sleep. Fifth, people who reduce their usual sleep time get less NREM1 and NREM2 sleep, but the duration of their slow-wave sleep remains about the same as before. Sixth, repeatedly waking individuals during REM sleep produces little increase in the sleepiness they experience the next day, whereas repeatedly waking individuals during slow-wave sleep has major effects. Circadian sleep cycles. The circadian rhythms. The world in which we live cycles from light to dark and back again once every 24 hours. Almost every physiological function in surface-dwelling animals displays some kind of circadian rhythmicity. The sleep-wake cycle is the most obvious. For example, most species display a regular circadian sleep-wake cycle. Humans take advantage of the light of the day to take care of their biological needs, and then they sleep for much of the night. In contrast, nocturnal animals such as rats sleep for much of the day and stay awake at night. The light-dark cycle is an important factor in the timing of most circadian rhythms. Environmental cues such as the light-dark cycle that entrain circadian rhythms are called Zeitgebers, a German word that means time givers. Free-running circadian sleep-wake cycles. What happens to sleep-wake cycles and other circadian rhythms in an environment that is devoid of Zeitgebers? Under conditions in which there are absolutely no temporal cues, humans and other animals maintain all of their circadian rhythms. Circadian rhythms in constant environments are said to be free-running rhythms. The duration of a free-running cycle is its free-running period. Even animals raised from birth in unchanging laboratory environments display highly regular free-running cycles. Circadian cycles thus do not appear to be learned. Sometimes, sleep-wake cycles and body temperature cycles can break away from one another so that they are out of phase. This is called internal desynchronization, and suggests that there is more than one circadian timing mechanism in the brain. The fact that such regularity is precisely maintained despite large day-to-day variations in physical and mental activity is strong support for circadian theories of sleep. Several studies have found a negative correlation between the length of a period of wakefulness and the length of the following period of sleep, even under free-running conditions. This is the opposite of what the recuperation theory would predict. It seems that we are programmed to go to sleep every 24 hours or so. If we stay awake longer than usual during a particular 24-hour cycle, there is less time left for sleep in that cycle. Drugs that affect sleep. Most drugs that influence sleep fall into two different classes. Hypnotic drugs are drugs that increase sleep. Anti-hypnotic drugs are drugs that reduce sleep. Hypnotic drugs. The benzodiazepines, example diazepam and clonazepam, which are GABA agonists, were developed and tested for the treatment of anxiety. Yet, they are some of the most commonly prescribed hypnotic medications. In the short term, they increase drowsiness, decrease the time it takes to fall asleep, reduce the number of awakenings during a night sleep, and increase total sleep time. Thus, they can be effective in the treatment of occasional difficulties in sleeping. Although benzodiazepines can be effective therapeutic hypnotic agents in the short term, their prescription for the treatment of chronic sleep difficulties, though common, is ill-advised. 5 complications are associated with the chronic use of benzodiazepines as hypnotic agents. First, tolerance develops to the hypnotic effects of benzodiazepines. Thus, patients must take larger and larger doses to maintain the drug's efficacy. Second, cessation of benzodiazepine therapy after chronic use causes insomnia or sleeplessness, which can exacerbate the very problem that the benzodiazepines were intended to correct. Third, benzodiazepines distort the normal pattern of sleep. They increase the duration of NREM2 sleep while actually decreasing the duration of both slow wave and REM sleep. Fourth, benzodiazepines lead to next-day drowsiness and increase the incidence of traffic accidents. And fifth, most troubling is that chronic use of benzodiazepines has been shown to substantially reduce life expectancy. Problems with the benzodiazepines have led to a search for other pharmacological agents with the same hypnotic effects as the benzodiazepines but fewer side effects. In the early 1990s, a new class of GABA agonists, the imidazopyridines, was marketed for the treatment of insomnia. It was claimed that they have fewer adverse side effects and less potential for addiction. Evidence that the raphe nuclei, which are serotonergic, play a role in sleep suggested that serotonergic drugs might be effective hypnotics. Efforts to demonstrate the hypnotic effects of such drugs have focused on 5-hydroxytryptophan, the precursor of serotonin, because 5-HTP, but not serotonin, readily passes through the blood-brain barrier. Injections of 5-HTP do reverse the insomnia produced in both cats and rats by the serotonin antagonist PCPA. However, they appear to be of no therapeutic benefit in the treatment of human insomnia. Antihypnotic drugs. The mechanisms of the following 3 classes of antihypnotic drugs are well understood: cocaine-derived stimulants, amphetamine-derived stimulants, and tricyclic antidepressants. The drugs in these 3 classes seem to promote wakefulness by boosting the activity of catecholamines, such as norepinephrine, epinephrine, and dopamine, by increasing the release into synapses, by blocking the reuptake from synapses, or both. Melatonin. Melatonin is a hormone synthesized from the neurotransmitter serotonin in the pineal gland. Sleep disorders. Insomnia includes all disorders of initiating and maintaining sleep. Hypersomnia includes disorders of excessive sleep or sleepiness. A third major class of sleep disorders includes all those disorders that are specifically related to REM sleep dysfunction. Both insomnia and hypersomnia are common symptoms of depression and bipolar disorders. Long-term reduction of nightly sleeps. 2 studies have been conducted. In each participant, a reduction in sleep time was associated with an increase in sleep efficiency, a decrease in the amount of time it took to fall asleep after going to bed, a decrease in the number of nighttime awakenings, and an increase in the proportion of slow-wave sleep. Long-term sleep reduction by napping. Most mammals and human infants display polyphasic sleep cycles, that is, they regularly sleep more than once a day. In contrast, adults display monophasic sleep cycles, that is, they sleep once a day. Long-term sleep reduction, a personal case study. The main positive was the added time to do things. The only negative of the experience was sleepiness. However, staying awake during the last hour of the author's bed, an hour during which the author usually engaged in sedentary activities such as reading, was at times a problem. That is when the author became personally familiar with the phenomenon of microsleeps, and it was then that the author required some assistance in order to stay awake. Each night of sleep became a highly satisfying but all too brief experience.